Imagine a world where aging doesn't have to mean a decline in cognitive abilities. Recent research has unveiled a fascinating connection between an iron-associated protein known as Ftl1 and cognitive decline in older mice, suggesting that targeting this protein could potentially reverse age-related cognitive impairments. This revelation challenges the traditional view of brain aging as an irreversible process, opening doors to new therapeutic strategies for enhancing cognitive function in the elderly. Understanding the relationship between brain aging and the molecular changes that contribute to cognitive decline is crucial for addressing the growing prevalence of neurodegenerative diseases. Studies have shown that cognitive dysfunction in older adults, even in the absence of neurodegenerative diseases, is often linked to a decline in neuronal function rather than cell death. This decline primarily occurs at the synaptic level, where communication between neurons is essential for learning and memory. To explore the molecular changes that occur in the aging brain, researchers focused on the hippocampus, a critical region for learning and memory that is particularly susceptible to aging. By employing advanced techniques like neuronal nuclei RNA sequencing, they identified significant changes in gene expression between young and aged mice. Specifically, they found 28 genes that were upregulated and 81 that were downregulated with age, with many of these genes related to synaptic processes. Among the identified genes, Ftl1 stood out due to its increased expression in aged mice. This protein plays a role in iron storage, and its elevated levels in the hippocampus of older mice correlated negatively with cognitive performance. This suggests that higher levels of Ftl1 may contribute to cognitive decline, making it a potential target for therapeutic intervention. To investigate the effects of Ftl1 on neuronal health, researchers conducted experiments using viral-mediated overexpression of Ftl1 in primary mouse neurons. The results were striking. Neurons with increased Ftl1 expression exhibited reduced neurite length and dendritic complexity, indicating impaired neuronal health. However, no significant changes in cell density or toxicity were observed, suggesting that the overexpression of Ftl1 specifically affects neuronal morphology and function. In a further step, the researchers aimed to mimic the age-related increase in Ftl1 within the adult hippocampus of young mice. They performed stereotaxic injections of a virus encoding Ftl1 into specific regions of the hippocampus. The outcomes were concerning. The overexpression of Ftl1 led to a decrease in synaptic proteins and impaired long-term potentiation, LTP, a key mechanism underlying learning and memory. Behavioral tests revealed that young mice with elevated Ftl1 levels showed no preference for novel objects, indicating cognitive impairment. Conversely, the researchers explored the effects of reducing Ftl1 levels in aged mice. By employing viral-mediated RNA interference to knock down Ftl1, they observed an increase in synaptic proteins and improvements in cognitive performance. Aged mice that underwent Ftl1 knockdown demonstrated enhanced memory capabilities, suggesting that targeting Ftl1 could reverse some of the cognitive deficits associated with aging. To delve deeper into the mechanisms at play, the researchers conducted transcriptomic analyses of hippocampal neurons from both young and aged mice. They identified significant changes in gene expression related to energy metabolism and synaptic processes, highlighting the potential role of metabolic dysfunction in age-related cognitive decline. Notably, they found that boosting metabolic function through NADH supplementation could mitigate the cognitive impairments associated with increased Ftl1 levels. In summary, this research sheds light on the complex interplay between iron metabolism, neuronal health, and cognitive function in aging. By targeting Ftl1, it may be possible to restore synaptic integrity and improve cognitive performance in older adults. This study not only enhances our understanding of the molecular underpinnings of cognitive aging, but also paves the way for innovative therapeutic strategies aimed at combating age-related cognitive decline. As we continue to explore these avenues, the potential for rejuvenating cognitive function in the elderly becomes an exciting prospect.